Dear viewers, this video is part 1 in the series on infections overview in the Medicine for Anyone in the YouTube channel. Infections. How many so-called citizens are living in our country? This question is a food for thought and humbling. Normal, healthy humans are colonized with about 50 trillion bacteria as well as countless viruses, fungi, and archaea. Taken together, these microorganisms outnumber human cells by about 10 times in the human body. Most of them are not visible to the naked eye. Dear listeners, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Now, coming back to discussion, bacteria were among the first life forms. Their size 0.4 to 3 micrometer are ubiquitous, mostly free-living organisms, often consisting of one biological cell. They constitute a large domain of prokaryotic, that means no nuclear membrane for packing in chromosomes, prokaryotic organisms. Viruses, size 0.02 to 0.25 micrometer. A virus is a submicroscopic infectious agent replicating only inside the living cells of an organism. Viruses infect all life forms, from animals and plants to microorganisms, including bacteria and archaea. Fungi, 2 to 10 micrometer in diameter and up to several centimeters in length. Most fungi grow as hyphae, which are cylindrical thread-like structures. A fungus is any member of the group of eukaryotic, that means with a well-defined nucleus and a nuclear membrane containing the chromosomes. Eukaryotic organisms that includes microorganisms such as yeast and molds, as well as the more familiar mushroom. Archaea, size 0.1 to 15 micrometers. Archaea is a domain of single-celled organisms. These organisms lack cell nuclei and are therefore prokaryotes. Morphology. Individual archaea range from 0.1 micrometer to over 15 micrometers in diameter and occur in various shapes, commonly as spheres, rods, spirals, or plates. Archaea can be found in the mud volcano area, among other places in Yellowstone, USA. Archaea are the most extreme of all extremophiles. Some kinds live in the frigid environments of Antarctica. Others live in the boiling acidic springs of Yellowstone, USA. Archaea are evolutionarily distinct from bacteria. Many archaea have been found living in extreme environments, for example at high pressures, salt concentrations, or temperatures. These types of organisms are called extremophiles. Archaea are everywhere, in deep sea vents, in salt flats, in ice, in sea, water, in soil, and in humans, and they deserve better publicity. Improved molecular detection tools led to the discovery of archaea in almost every habitat, including soil, oceans, and marshlands. Archaea are particularly numerous in the oceans, and the archaea in the plankton may be one of the most abundant groups of organisms on the planet. Archaea are a major part of Earth's life. This picture shows relative sizes of the organisms. On the left is the range of visibility for each type of object, human eye and the different microscopes. On the left are the different organisms and their range of sizes. See figure relative sizes of organisms. Human heart, for comparison, is 10 cm wide. 1 meter is 1000 millimeters. 1 millimeter is 1000 micrometers. 1 micrometer is 1000 nanometers. The major reservoir of these microbes is the gastrointestinal tract, but substantial numbers of microbes live in the female genital tract, the oral cavity, and the nasopharynx. There is increasing interest in the skin and lungs as sites where microbial colonization might be highly relevant to the biology and disease susceptibility of the host. These commensals, that is, they don't do any harm to humans and even may be useful. These commensal organisms provide the host with myriad benefits, from aiding in metabolism to shaping the immune system. With regard to infectious diseases, the vast majority of infections are caused by organisms that are part of the normal microbiota. Example, Staphylococcus aureus, Streptococcus pneumoniae, Pseudomonas aeruginosa, with relatively few infections due to organisms that are strictly pathogens. Example, Neisseria gonorrhoeae, causing gonorrhea, a sexually transmitted disease, rabies virus causing rabies. Microbes, our ancestors. The basic plan of the microbes. To make something recognizable as a living organism, you would need two basic sets of machinery: metabolic to generate energy, and hereditary to produce, reproduce, and create the next generation. Without heredity, your construction would remain a one-off, never to be replaced when it wore out. Without its own metabolism, it would be inert. For metabolism, nature has exploited a variety of chemical chain reactions or pathways, some using oxygen and some not, and there are bacteria that specialize in each of them. But there is only one hereditary mechanism, the one we are familiar with, using the nucleotides DNA and RNA. See the gene control of cell function in Medicine for Anyone in the YouTube channel. It now seems certain that RNA was the first molecule of heredity, so it evolved all the essential methods for storing and expressing genetic information before DNA came into the scene. There is great resemblance between the ribosomal RNAs of all living creatures. There are organisms which have the RNA or DNA, but no metabolic equipment of their own. These are the viruses, and some of them are undoubtedly pathogens. Metabolically active chemicals and potentially reproductive nucleotides floating around freely still do not constitute a living organism. For this, they need to be held together in some reasonably stable structure, and this happened when the first cell membrane evolved, to be followed later by the cell wall with its increased strength and resistance to external conditions. And this is essentially the bacterial type of cell. This figure is a typical bacterial cell, a rod-shaped bacillus with a cell wall, cytoplasm, chromosomes without a nuclear membrane, RNA with pili and flagellum. Capsules and fimbriae are sometimes present. A typical bacterial cell, a rod-shaped bacillus, and its parts were shown earlier. A cell with a wall and a membrane containing enzymes to speed up the metabolic reactions, which would otherwise be painfully slow, plus DNA, its bases strung together on a chromosome, capable of replicating itself and arranged as genes that, with the help of RNA, form proteins able to make more walls, membranes, and enzymes. It is a magnificent creation, fully capable of independent existence and self-replication for billions of years, and has expanded into every possible ecological niche in the world. Bacterial reproduction is about as simple as you can get. The cell doubles in size and splits in two. The progeny are normally identical but do not remain so forever because bacterial DNA is subject to change by mutation, and there are even mechanisms for exchanging DNA between bacteria. This can come in useful when they are under attack, for instance by antibiotics. Bacterial cells come in various shapes and sizes: spherical, rod shaped, spiral, etc., and are named according to their appearance under the microscope. This picture shows some common shapes of bacteria. Cocci are spherical, arranged in clusters or in chains. Bacilli are rod shaped, may or may not have spores. Spirochetes are spiral. Actinomycetes are branched filaments. Vibrios are comma shaped. More precise classification depends on the pattern of staining with various dyes, analysis of surface structures, and eventually DNA sequencing. Further additions. The bacterial type of cell is known as prokaryotic, meaning pre-nucleated, to distinguish it from the later type of eukaryotic, that is well-nucleated cell, of which all other structures, from the single-celled amoeba to large multinuclear plants and animals, are built. The first novel feature in the eukaryotes was the nucleus, in which the majority of the DNA is packaged, now divided up into a number of different chromosomes, 23 pairs of chromosomes in the case of humans. But the eukaryotic cells did not stop there, because it emerged that the prokaryotes still had something to offer by taking up residence inside eukaryotes, a process known as endosymbiosis, that is, inside together living, a good example of useful parasitism. This must have occurred at least twice. An oxygen breathing bacterium settled down as a permanent cell component called the mitochondrion, providing a compact source of energy to animal cells, while a chlorophyll containing green bacterium breathing carbon dioxide and producing oxygen gave rise to the chloroplast found in all plant cells. So it was the bacteria that decided the future of the entire plant and animal kingdoms. Not a bad achievement for a microscopic organism. So, dear viewers, this is the end of video part 1 in the 4-part series on infections overview in the Medicine for Anyone in the YouTube channel. Thank you for watching.